Hello friends, today's presentation topic is about transgenic animals. Introduction. A transgenic animal is one that carries a foreign gene that has been deliberately inserted into its genome. Transgenesis is a process by which mixing up of genes takes place. Foreign genes are inserted into the germ line of the animal so it can be transmitted to the progeny. Transgenic technology has led to the development of fishes, livestock, and other animals which altered genetic profiles which are useful to mankind. First transgenic animal was a super mouse created by Ralph Brinster, U. Pennsylvania, and Richard Palmiter, University of Washington in 1982. It was created by inserting a human growth hormone gene in mouse genome. The offspring was much larger than the parents as shown in the figure. Mouse. It is used commonly in the transgenic experiments. And some of the other animals include pig, goat, cow, sheep, fish, etc. Production of transgenic animals. The methodology. Step 1. Construction of a transgene. Transgene made up of three parts: promoter, gene to be expressed, termination sequence. Step 2. Introduction of foreign gene into the animal. Pronuclear microinjection method, embryonic stem cell method. The first, microinjection method. A female animal is superovulated and eggs are collected. The eggs are fertilized in vitro. The transgene containing solution is injected into the male pronucleus using a micropipette. As you can see the picture. And eggs with the transgenes are kept overnight in an incubator to develop to a two-cell stage. The eggs are then implanted into the uterus of a pseudo-pregnant female. It means female which has been mated with a vasectomized male the previous night. Second one, embryonic stem cell method. Transgenic animals can be created by manipulating embryonic stem cells. Embryonic stem cells are obtained from the inner cell mass of a blastocyst. You can see the figure of a blastocyst. Transgene is incorporated into the embryonic stem cell by microinjection, by a retrovirus, by electroporation. Transgenic stem cells are grown in vitro. Then they are inserted into a blastocyst and implanted into a host's uterus to grow normally. And this is the microinjection which describes the microinjection. And step 3, screening for transgenic positives. Transgenic progenies are screened by PCR to examine the site of incorporation of the gene. Some transgenes may not be expressed if integrated into a transcriptionally inactive site. Further, animal breeding is done to obtain maximal expression. Heterozygous offsprings are mated to form homozygous strains. Method 1 and method 2 are done in this way as shown. Problems. Multiple insertion. Too much proteins, insertion into an essential gene, lethality, insertion into a gene leading to gene silencing, insertion into a different area can affect the gene regulation. Some examples of transgenic animals. The first one, transgenic fish. Superfish, increased growth and size. Growth hormone gene inserted into fertilized egg. Transgenic salmon grows about 10 to 11 times faster than normal fish as shown in the figure. Glowfish, GM freshwater zebrafish, Danio rareo, produced by integrating a fluorescent protein gene from jellyfish into embryo of fish. Next, transgenic mouse. Alzheimer's mouse. In the brain of Alzheimer's patients, dead nerve cells are entangled in a protein called amyloid. Mouse made by introducing amyloid precursor gene into fertilized egg of mice. Oncomouse. Mouse model to study cancer, made by inserting activated oncogenes. Smart mouse. Biological models engineered to overexpress NR2B receptor in the synaptic pathway. This makes the mice learn faster like juveniles throughout their lives. Transgenic pig, Enviro pig. Pigs have trouble fully digesting a compound known as phytate found in many cereal grains used to feed them. Transgenic pigs are created by introducing phytase gene of E. coli. As these pigs are unable to digest the compound phytate, so they are genetically modified by phytase enzyme is thus produced in the salivary gland of pig. It degrades indigestible phytate with the release of phosphate that is readily digested by pigs. In this way, these pigs can digest the phytate compound. Pig for organ transplant. Pigs with human genes in order to decrease the chance of organ rejection by human body. Next, transgenic livestock. Bioreactors whose cells have been engineered to synthesize marketable proteins, more economical than producing desired protein in cell culture. Transgenic cattle. Transgenic cows are made to produce proteins, lactoferrin, and interferons in their milk. Prion-free cows resistant to mad cow disease. Transgenic sheep for good quality wool production. Sheep are modified genetically for the wool production. Next, transgenic goat. Goats that could express tissue plasminogen activator, antithrombin 3, spider silk, etc. in milk. Transgenic rabbit. Alba, the EGFP, enhanced green fluorescent protein bunny, created in 2000 as a transgenic artwork. Transgenic monkey. And I was the first transgenic monkey born in 2000. And I stands for inserted DNA, spelled backwards. An engineered virus was used to insert the harmless gene for green fluorescent protein into I's rhesus genome. I proves that transgenic primates can be created and can express a foreign gene delivered into their genome. Importance of transgenic animals. Medical importance. Disease model, bioreactors for pharmaceuticals, xenotransplantation. Agriculture importance. Disease resistant animals for improving quality and quantity of milk, meat, eggs and wool production. Industrial importance. Toxicity sensitive transgenic animals to test chemicals. Spider silk in milk of goat. Issues related to transgenic technology. Blurring the lines between species by creating transgenic combinations. There may be health risks associated with transgenics. There may be long-term effects on the environment which transgenic animals are released into the field. Various bioethicists argue that this is wrong to create animals that would suffer as a result of genetic alteration. Recent trends. Transgenic goats engineered to produce human breast milk. University of California scientists created the transgenic goats by transferring human genes for breast milk enzymes and proteins into goat embryos. Produce 60% of the lysozyme and lactoferrin found in human mother's milk. For babies of mothers who aren't present or can't nurse them, milk from these transgenic goats could provide the next best alternative. Bioluminescent transgenic mouse model for study mammary gland tumor development. Research done by Cancer Biology and Therapeutics Group, Ireland. Conclusion. Transgenic technology is a field that is under constant evolution. Many transgenic animals have been successfully created for a variety of purposes and the prospects are enormous. It holds great potential in many fields including agriculture, medicine and industry. With proper research and careful use, the transgenic animals can go a long way in solving several problems for which science doesn't have a solution till now. Thank you. Like, share and comment.